Good morning, everyone. So the next 15 minutes, I'm going to walk through really how we, an approach to modulate the immunopeptidome using this thing called ERAP1 inhibition, and how we've shown that this plays out as a therapeutic in an autoimmune condition called axial spondyloarthritis, and particularly these patients, these axial spondyloarthritis patients that have this HLA-B 2705. Oh, sorry, that was my title. So a little bit of background on Greywolf, just before I get started. So we are a clinical stage biotech company, and our scientific approach really is to target this signal 1 component of T-cell activation. So T-cell activation requiring a chain of 3 signals, where most current therapeutic approaches are focused on the latter stages in this signaling cascade. But what we've really shown is that by manipulating signal 1, which is antigen presentation, we can impact the visibility of cells to the immune system. And this plays out in oncology, but it also plays out in autoimmunity as well. So ERAP1, which is the enzyme that we inhibit, is an aminopeptidase in the endoplasmic reticulum. And its primary function is in peptide substrate cleavage prior to presentation on MHC1. So in any cell, you will have proteins. These proteins will be degraded by the proteasome. Those peptide chains will then be transported into the ER, whereby a proportion of which will undergo cleavage, and then they'll subsequently be loaded onto MHC and presented to the immune system. So in the context of autoimmunity, the same happens for those autoantigens as does normal peptides. So they undergo some ERAP1 dependent cleavage, and they're in particular in axial spondyloarthritis, they are presented on B2705. To these autoreactive T cells. So that's what happens under functional ERAP1 conditions, and then our approach is really to turn ERAP off, and by turning ERAP off, you prevent this peptide substrate cleavage, and you block the presentation by changing the antigen repertoire at the cell surface. So you ablate this autoreactive T cell response. So axial spondyloarthritis is a predominantly CD8 T cell driven disease. And really what this schematic depicts here is just the lead hypothesis around the early event of this cross-recognition of self-autoantigens. So there is an initial microbial event, normally an infection in these patients. This microbial peptide is presented on HLA-B 27. It is then cross-recognized as a self-antigen by T cells in these patients, and this is where really autoimmunity starts. And I think just to highlight here, there's some really compelling genetics. So if you are an individual that carries a particular variant in ERAP1, and this particular variant is a hypermorphic variant, so you still have the ERAP1 enzyme, but it's just hypermorphic, so it's inactive, you are significantly protected against developing axial spondyloarthritis, and the association there is about 10 to the minus 41. So not only is that the case with ERAP1, but there is also an epistatic relationship with HLA-B 2705 in these individuals, and that association is 10 to the minus 27. So if you have a hypermorphic ERAP1 in those patients that are HLA-B 2705 negative, you are incredibly protected from this disease. So we really wanted to understand where the link was between ERAP1, the peptide presentation of these autoantigens in these HLA-B 2705 individuals. And to sort of tackle this from an exploratory perspective, we looked into 3 model systems using immunopeptidomics. So 3 model systems, PBMCs, where all of the PBMC donors had endogenous expression of HLA-B 2705, SW1353 cells, where we had to transduce in HLA-B 2705 allele, and then HT1080 cells, which do have endogenous HLA-B 2705, but it wasn't quite to the level we needed it at, so we also transduced that in as well. And then we treat all of these cells with our inhibitor and generate pellets that we send to Biognosis, and then perform DIA-based immunopeptidomics. And all of these are searched in Spectronaut. So I think just on the right-hand side, just to go over this briefly, and I guess really just to say top graph on the right, I broke the axis just because we have 3 independent PBMCs where only the HLA-B 2705 is shared, so we naturally get many more peptide IDs there, so we get about 41,000. But in the SW1353 lines, we're getting about 22,000 peptides and about about 14,000 in the HT1080. And then the bottom right really just illustrates how many of those are relevant to the model system we're studying. So we have about 9,000 HLA-B 2705 peptides to study in the PBMCs, 6,000 in SW1353, and about 3.5 in HT1080. So we started to try and characterize why ERAP1 inhibition may play out as a therapeutic in abrogating presentation of these autoantigens. And really just starting on the left-hand side here for these three volcano plots, what is very clear to see, and we know is a very typical biomarker of engaging ERAP1 as a target, because you are inhibiting the cleavage of these peptide substrates in the ER, you drive the presentation of longer peptides at the cell surface. So you drive presentation of proportionally longer peptides, and that is what is depicted here. So what you're looking at in these volcano plots, on the far left of these 7 to 9 mers that are mainly illustrated in purple, so you're mainly down-regulating 7 to 9 mers, and you're predominantly up-regulating these 10 to 14 mer peptides. And then the right-hand side really just depicts that in a length-frequency plot. So if you look at the length distribution of those up versus down-regulated peptides, you can see the purple bar is what you would typically expect to be a very normal MHC1 profile, but you can see when you treat with a drug, you decrease those 9 mers and increase the frequency of longer peptides. And then this effect really is incredibly pronounced on HLA-B 2705. So the top graph being all alleles in that particular cell line, and the bottom being the effect of that length frequency on B2705 itself. So we have a really large effect on 9 mers here, and we really drive the presentation of longer peptides on this allele. So we thought that was maybe one piece of the puzzle. So maybe B2705 as an allele is just incredibly permissive to presenting these longer peptides. And we'd done some in silico work to try and see if that was the case. So we took these up and down regulated peptides on this allele, and we just looked with NetMHC-PAN to predict their binding affinity. And I know there's obviously a lot of caveats with that approach, but we were really looking for a hypothesis lead to go and validate further. But what you can see quite interestingly is that on the right-hand side, if you follow those purple bars from 10, 11, 12, 13, and 14 mers, you can see that they are actually all predicted to be stronger binders relative to any of those other alleles in that cell line. So there is something about B2705 that we think that will just readily accommodate these longer alleles, but that's not the entirety of the story here. So if you then look at the motifs of those differentially expressed peptides, so the top left graph here being those down-regulated 9 mers, you can see there's quite a strong restriction for arginine and glutamine at the P2 position. If you then take the upregulated peptides for the 10 mers and the 11 mers, and this also plays out for 12, 13, and 14, but I didn't think fitting 5 graphs onto one slide was really going to be that constructive. You can see there is a really strong preferential retention of this arginine at P2. So not only are these peptides just longer and just stochastically random, you have to retain this P2 position as well for presentation. So we'd map this back then on the right-hand side to the context of these autoantigens. So there is a paper out in Nature that had used the yeast display system, and they'd screened these HLA-B 2705 peptides for autoreactivity against axial spondyloarthritis patients' TCRs. And they'd come up with this list of 9 putative autoantigens. And you can see that all of those 9 mers, those putative autoantigens, will have that arginine or that glutamine at P2. So then if you think about this in the context of what our drug is doing, so in the context of ERAP1 inhibition, you will extend the N-terminus of these peptides. And if you take the N-terminal extension of these peptides, what actually happens is you remove that P2 preference completely. So we think this is not just a length thing, but it's a length and loss of specificity, which I guess makes sense from a binding perspective. So our hypothesis here really was that N-terminal extension of these autoantigens would just ablate their binding to HLA-B 2705. And this is the sort of hypothesis that we generated that we then went on to further test experimentally. So we show in a Mauditov system that you can get very, very potent inhibition of mature autoantigen production for a number of these autoantigens. So we didn't test all of them, but these are 3 that we tested, and you would get picomolar affinity against these antigens with our inhibitor. And then interestingly, to sort of prove this hypothesis about N-terminal extension and loss of binding specificity, we take a mature autoantigen here with this arginine at P2, and test it in a purified HLA-B 2705 binding assay, where you're essentially asking, relative to a labeled reference peptide, how much can you displace based on binding affinity? And what is really interesting to see is that you take the 9-mer, it has high affinity for B2705. You then extend, so you generate a synthetic peptide with an N-terminal extension, just one single amino acid, and you decrease the binding affinity by 60-fold. You do 2 amino acids, and you decrease it by 120-fold, and 3 will get you to a 170-fold decrease in binding affinity for this particular autoantigen. And then I think just as a positive control here to show how important that P2 position is on this particular allele, you can take the 9-mer and just replace the P2 arginine with an alanine, and you also get the same effect as extending it by +2. So we've really shown here, I think, that by N-terminally extending these peptides, they're just not making it, in the world of competitive dynamics in the endoplasmic reticulum, they're just not making it back onto the allele. And that's what hopefully we've illustrated here as well. So we now take a selection of these pathogenic TCRs, so these TCRs that have been shown in the paper to be cross-reactive with these autoantigens. And we first validated that cross-reactivity existed, so we made a JERCAT-based system that expressed these TCRs, and we also overexpressed this autoantigen as a 14-mer mini-gene. So a 14-mer mini-gene that was tagged to go directly into the endoplasmic reticulum. So what you can see, and what is illustrated by green here, is just that all of these TCRs recognize the mature autoantigen. So we know that is a fact. And then again on the right-hand side, if you take this system and you treat these cells in this co-culture with 715, you can decrease the activity of all 4 of these TCRs against this autopeptide. So this is, again, just to reiterate, this is 14-mer into the ER, under normal functional conditions, is cleaved by ERAP to produce that mature 9-mer autoantigen, obviously then cross-reacts with T cells, and then you inhibit that process and you get no presentation at the cell surface, and you can ablate this T cell response. And not only can we do that, we can do that in a dose-dependent manner as well. So the graph on the bottom here just shows that we can do a titration of our drug and get a really nice dose response. So I think just to wrap all of that up, I guess quite a lot of different data. We used immunopeptidomics to define a hypothesis and do some exploratory work to try and understand why, based on the genetics of this disease, ERAP1 inhibition could play therapeutically in this particular condition. And based on that hypothesis, we went on to do some enzymology and binding assays, and we show that ERAP1 inhibition not only prevents the mature autoantigen production, but it does so by the N-terminal extension of that peptide ablating binding to B2705. And then the final piece of the story was really the T cell activation. So if you create a system where T cells recognize this peptide, you can completely block that recognition by inhibiting ERAP1 in this system. And just to say that we opened a phase 1/2 clinical trial for this disease earlier in the year for HLA-B 2705 positive patients. And I think just with that, I'll wrap up and say that this is by no means all me. It's absolutely a team sport to develop drugs. And I think just to thank Biognosis again, but also thank all of the people in this picture who help and contribute to this work.